Okay, cilia and flagella. Tail-like structure, one flagellum, many flagella. And hair-like structure, moving hair. Hair-like structure, we call one cilium, many cilia. So they are composed of microtubules. Microtubules control the beating of flagella and cilia. Microtubule containing extensions that project from some cell. So that means some cell, the flagella or flagellum, they extend out from the one cell. Cilia, same. Cilia, usually they covering up the whole cell. And many cilia, they covering up the whole cell. And the cilia and flagella differ in their beating patterns. This one showing you the beating patterns of flagellum. So that's the typical animal sperm cell. So they beating up to swim. And the hair-like structure, we call they are cilia. So cilia motion, they moving back and forth like that. So that's the beating, another type of beating. That's the overall structure of flagellum. So flagellum structures, the upper side and downside different. So we can see cross-section of motile cilium or flagellum, that's the same. Structures are same. And the downside to the cell side, So flagellum, it is inserted into the cell. To the cell side, the down bottom, we can see basal body. So composed of 9 triplet of microtubule. And we're going to discuss detail in next picture. This one is showing you the basal body part. So microtubule connected into the inside the cell. So inside the cell, the structure gonna get different, become a basal body. So that's why that's the base of the flagellum or cilium. So that's the basal foundation structure we call basal body. Cilia and flagella share a common structures. A core of microtubule sheathed by the plasma membrane. So they are surrounded by plasma membrane. Cilia and flagella, they are surrounded by plasma membrane. That means that's the part of the cytoplasm. A basal body that anchors the cilium or flagellum into the main body of the cell. A motor protein called dynein, which drives the bending movements of a cilium or flagellum. This one showing you the structure of cilium or flagellum. And we can see left-hand side picture, that's the cross-section of motile cilium. And we can see the microtubule doublet. Doublet means 2 microtubules they put together, make doublet. So microtubule doublet, we have at the left-hand side, the picture at the ring. We can see the ring, and we can see those, how many doublet? 1, 2, 3, 4, 5, 6, 7, 8, 9 doublet. And they arranged in ring outside, and inside at the center we can see single, 1, 2 single microtubule we can see. And when we look at right hand side picture, one doublet of microtubule, from that one doublet the red color thing they extend out, yeah, like a hand, yeah. So like Y, like a hand, they extend out, yeah. So 1, 2, the dynein, we call that motor protein has a nickname as dynein arms, because those motor protein they act like human arms, yeah. How? They grabbing like hand, yeah. So from a one doublet it can extend out and grabbing the next doublet. So what they do, after grabbing with the dynein arm, they push downside or push upside for that next doublet. So that's why when they coordinate their motion with the dynein protein, so at the right hand side, that microtubule doublets dynein arm grabbing the next and push down, and the next dynein arm grabbing the next one and push down, down, down. Then what's going to happen? Because when they coordinate their motion, they're going to gradually push down, down, down to the right hand side. Yeah. Then what's going to happen? Because when they gradually push down to the left hand side, the whole structure of the cilium, they're going to get bent from a right hand side to left hand side, bend. And after that, when they coordinate their motion opposite, then whole structure, the cilium gonna bend from left hand side to right hand side. So the result, they can move from one side to the other and the other side to one side. They can move back and forth, back and forth, back and forth like that. That motion we call, that's the beating, yeah. Beating, B-E-A-T, beating, beating, moving back and forth like that because of the dynein arms, that motor proteins function. That's very important for the bending. So that's why cilium and flagellum, basically their motion is same, beating back and forth like that. Those cilium or flagellum, they are connected into the main body of the cell. Their structure for the connection is different. Yeah? We call that the basal body. It is composed of triplet of microtubule. How many? 9. So make a ring shape like that. That's it. Yeah. So that's we call that's the basal body. So at the basement of flagellum and cilium, we can find basal body, and we can find where somewhere else. So that's the centriole. Yeah. Centriole, their structure exactly same as like this. That's the basal body. That's the centriole structure. Okay, as we discussed briefly already, how dynein walking moves flagella and cilia. Dynein arms alternately grab, move, and release the outer microtubules, and protein cross-links limit sliding. Forces exerted by dynein arms cause doublets to curve, bending the cilium or flagellum. And next one we're gonna discuss over that's the microfilaments or actin filaments. Microfilaments are solid rod about 7 nanometers in diameter, built as a twisted double chain of actin subunits. The structural role of microfilaments is to bear tension, resisting pulling forces within the cell. They form a three-dimensional network called the cortex just inside the plasma membrane to help support the cell shape. And bundles of microfilaments make up the core of microvilli of intestinal cells. This one showing you the structure of microvilli composed of microfilament, actin filament. And at the basement, we can see that's the intermediate filaments for the microvilli. Microfilaments that function in cellular motility contain the protein myosin in addition to actin filaments. In muscle cells, thousands of actin filaments are arranged parallel to one another. Thicker filaments composed of myosin interdigitate with the thinner actin fibers. As I discussed already, when we contract our muscle cell, the actin filament and myosin filament, they overlap each other. So the muscle gonna get contracted. And when we relax the muscle, they're gonna separate back again. So that's how the actin filament and myosin filament interacted each other. This one showing the actin filaments network. They are responsible for amoeboid movement. So with the actin network, they extend out pseudopodium like that. And those actin filaments movement, they are responsible for cytoplasmic streaming in plant cell or cyclosis. So localized contraction brought about by actin and myosin also drive the amoeboid movement, and cells crawl along the surface by extending pseudopodia and moving towards them. Cytoplasmic streaming is a circular flow of cytoplasm within cells. This streaming speeds distribution of material within the cell. In plant, actin-myosin interactions and sol-gel transformations drive cytoplasmic streaming. Intermediate filaments range in between. Never mind, that's real 8 to 12 nanometers something, never mind. That's larger than microfilaments, but smaller than microtubule, intermediate. They support cell shape and fix organelles in place, so tied up to cytoskeleton with intermediate filaments. Intermediate filaments are more permanent cytoskeleton fixtures than the other two classes. Outside the plasma membrane, we call extracellular. So extracellular components and connections between cells help coordinate cellular activities. Most cells synthesize and secrete material that are external to the plasma membrane, so extracellular. These extracellular structures are involved in a great many cellular functions. Cell walls of plants. The cell wall is an extracellular structure that distinguishes plant cells from animal cells. Prokaryotes, fungi, and some unicellular eukaryotes also have cell walls. The cell wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. Plant cell walls are made up of cellulose fiber embedded in other polysaccharides and protein. Plant cell walls may have multiple layers. So the first, they developed primary cell wall that's relatively thin and flexible. So when new cell born, they produce first. That's why we call that's a primary cell wall. And when one cell with the cell wall, the other cell with the cell wall, when they connect it together with middle lamella, that's the thin layer between primary walls of adjacent cell. And when we call secondary cell wall, some cells they develop secondary cell wall. So after they produce primary cell wall, some plant cells they can produce secondary. So that's why they produce inside the cell and secrete out. That's why they're gonna get formed inside the primary cell wall. That's the secondary cell wall. So that added between the plasma membrane and the primary cell wall. Usually they are very strong and thick. And plasmodesmata are channels between adjacent plant cells. Sometimes plant cell to another plant cell, they have some difficulty for their communication because they are separated by strong cell wall. But there are some tiny channels connecting one cytoplasm through the cell wall into next cytoplasm of the plant cell. Those kind of tiny channel connecting one cytoplasm to another through that plant cell wall, we call that the plasmodesmata. One plasmodesma, more than one plasmodesmata. Actually, desma means it's like a bridge, desma, and plasmo indicating cytoplasm. So it's like a bridge connecting one cytoplasm to another cytoplasm through that cell wall. That's why name is plasmodesma or plasmodesmata. This one showing you one cell with a cell wall covering and another cell with the cell wall covering the connecting line. We call that the middle lamella. So with our middle lamella, one cell and another cell connected together. So with the middle lamella and next layer, we can see that's the primary cell wall, kind of thin and flexible. And some plant cell, they can produce secondary cell wall and secrete out inside the primary cell wall. So we can see those kind of thick secondary cell wall for some plant cell, not all, but some plant cell. And down below, they showing the diagram for plasmodesmata. That's the tiny channel connecting one cytoplasm to another through that plant cell wall we call plasmodesma or plasmodesmata. How about animal cell? Animal cell structure outside the plasma membrane, we call that the extracellular matrix, ECM. Animal cells lack cell wall, but are covered by an elaborate extracellular matrix, ECM. The ECM is made up of glycoprotein such as collagen. Collagen, that's a huge protein, and proteoglycan and fibronectin protein. And ECM proteins bind to receptor protein in the plasma membrane called integrins. This one showing you the model for extracellular matrix. So we can see plasma membrane, and through the plasma membrane we can see integrin protein. So integrin protein, they are embedded through that plasma membrane we call integrin protein. And outside the plasma membrane, we can see extracellular matrix composed of mostly polysaccharide plus protein complex, like a collagen. That's a huge protein that's very important component in extracellular matrix for animal cell. So outside the ECM, it is like a network, yeah. It's like, so they form a network like this, and connecting plasma membrane into the network of extracellular matrix, that's the fibronectin. Yeah? So they connecting plasma membrane to outside extracellular matrix, fibronectin. So the ECM has an influential role in the lives of cells. ECM can regulate a cell's behavior by communicating with the cell through integrins. The ECM around a cell can influence the activity of genes in the nucleus. Mechanical signaling may occur through cytoskeletal changes that trigger chemical signals in the cell. So, inside some signal, they go through inside the cell through the cytoskeleton, and cytoskeleton is connected into integrin, and outside fibronectin go out into the ECM. So they are all connected, inside, outside, connected together. Okay, tissue, that's the group of same type of cell, we call that the tissue. So cells are connected with the next cell, they get together. Then how they get together, we call that the cell junctions. So neighboring cells in tissues, organs, or organ systems often adhere, interact, and communicate through direct physical contact. Not exactly cell junction, but plasmodesmata in plant cells are important for their communication and transportation. So plasmodesmata are channels that populate plant cell wall. So they're connecting together through the cell wall. Through plasmodesmata, water and small solute, and sometimes proteins and RNA can pass from cell to cell. So the plasmodesmata, that's very small, tiny channel. That's why only the water and small solute they can go pass through the plasmodesmata. Big molecule is kind of difficult to go pass through. Okay, for the cell junction for animal cells, we have 1, 2, 3 types. So 3 types of cell junctions are common in epithelial tissues, skin tissue for the animal. That's the epithelial. Tight junction means one cell to another, they join together without any space in between cells. So at tight junction, membranes of neighboring cells are pressed together, preventing leakages of extracellular fluid. So they don't allow any space in between cells. That's what we call tight junction. They join, they, I mean, they join together tightly, so without any space. However, there's more some. Desmo means another meaning of bridge. Desmo, bridge. So desmosome, they connected two cells like a bridge. They allow some space. So in other words, desmosome we call sometimes anchoring junction. Fasten cells together into strong sheaths, but they allow some space in between two cells. And the third one, that's the gap junction. They allow specialized channel for transportation, like plasmodesmata. That's the, in case for the animal, we call that's the gap junction. So in other words, we call that's the communicating junctions. Provide the cytoplasmic channel between adjacent cells. This one showing 3 types of cell junction for animal. So we can see tight junction and desmosome and gap junction. As we discussed, one cell to another, they connected together without any space in between. We call that the tight junction. So like our skin tissue or inside our intestine, inner surface, our intestine, they have to join together tightly without any space. If they leaks out, they're gonna get in trouble. So the skin tissue, that's the same. That's the desmosome. So one cell to another cell, they join together strongly, but they allow some space in between, like a bridge. So it's desmosome. That's the gap junction. They allow specialized channel from one cell to another. So through the channel, they can do communication and transportation, like plasmodesmata gap junctions. So the cell is composed of many subcellular structures. That's the basic unit for living activity. But the cell, that's a living unit greater than the sum of its parts, sum of its components. Cells rely on the integration of structures and organelles in order to function. For example, a macrophage's ability to destroy bacteria involves the whole cell coordinating components such as the cytoskeleton, lysosome, and plasma membrane. So they have to cooperate together. They show some integration. All those parts or components, they have to cooperate each other. So that's the more than, greater than, sum of is just components. Okay, that's it for this chapter. Thank you.